Hi, today I'm talking about Helicobacter pylori. At the very beginning, we will know its scientific classification. The domain is bacteria, the phylum is Campylobacteriota, the class is Campylobacteria, the order is Campylobacterialis, the family is Helicobacteriaceae, the genus is Helicobacter, and finally the species is H. pylori. Next, the morphology. Helicobacter pylori is a gram-negative bacterium measuring 2 to 4 micrometer in length and 0.5 to 1 micrometer in width. Usually spiral shaped, the bacterium can appear as a rod. It has 2 to 6 uniporal sheathed flagella of approximately 3 micrometer in length, which often carry a distinctive bulb at the end. The flagella confer motility and allow rapid movement in viscous solutions. Next, I'd like to present some facts as an introduction. H. pylori was discovered in the late 19th and early 20th centuries. The Australian novelist Dr. Barry J. Marshall and Dr. J. Robin Warren first discovered H. pylori in 1982. H. pylori infection was considered as the causative agent of gastritis, peptic ulcer disease, mucosa-associated lymphoid tissue lymphoma, and gastric cancer. About half of the world's population is estimated to be affected by Helicobacter pylori infection. 94% of patients with gastric ulcers and 98% of patients with duodenal ulcers have an infection of H. pylori. And finally, the most effective treatment appears to be bismuth-based quadral therapy, and the second treatment is levofloxacin-based triple therapy. For the transmission, there is no accurate knowledge of the mode of transmission of H. pylori. Therefore, it has been hypothesized that H. pylori is transmitted through oral-to-oral and fecal-to-oral routes associated with contaminated food and water. Also, H. pylori has several adaptations. First, low pH. H. pylori can survive short exposure to a pH of 4. Second, low O2. H. pylori can survive in an environment with a low level of oxygen. Third, shape switch. H. pylori can navigate through two distinct shapes, rod shape and cocoid shape. Finally, they survive over other unsuitable environmental conditions like the temperature change, the long intervals among meals, and antibiotic treatment. Interestingly, H. pylori colonization and the establishment of diseases and infection depend on four main stages. First, the acidic environmental adaptation of the gastric mucosa. Second, moving towards and penetrating the epithelial cell barrier. Third, attaching to receptors. And finally, tissue harm and other damaging health effects. Therefore, to successfully establish an infection and colonize the host, H. pylori must first survive the acidic stomach, attach to the surface of the host cells, and release toxins that cause tissue damage. After establishing an infection, H. pylori releases damaging tissue toxins, and some of the main effector proteins toxins that H. pylori released are blood group antigen binding adhesion, outer inflammatory protein, outer membrane protein, outer membrane vesicles vacuolating cytotoxins, a cytotoxin associated gene product, high temperature requirement A, neutrophil activating protein A, and sialic acid binding adhesions. In quick, I will mention the most known virulence factors: flagella, adhesions, outer membrane proteins, shape switch, urease production, vacuolating cytotoxins, VAC-A, cytotoxin associated gene product KA, type 3 and 5 secretion systems induced by contact with epithelium gene A, ICE-A, duodenal ulcer promoting gene DUP-A, and finally the high temperature requirement A, HTRA. Next, laboratory diagnostics. There is two type of methods. First one, the invasive methods, which included endoscopy, rapid urease test, histology, culture, and molecular methods. The second type of methods is the non-invasive methods, which include urea breath test, SAT, serology, and emerging diagnostic method. Next, the treatment strategies. Treatment guidelines for H. pylori infection are either a triple or quadruple therapy, depending on the patient background, geographical location, and socioeconomic status. The most broadly prescribed treatment for H. pylori infections is triple therapy, which consists of clarithromycin, amoxicillin, and a proton pump inhibitor (PPI) given over 14 days in the United States, Europe, and Canada. Recent clinical guidelines advocate for 14 days of quadruple therapy, which can be bismuth-based or concomitant therapy containing 3 broad-spectrum antibiotics and a PPI as first-line treatment. These regimens offer perfect eradication rates of approximately 90%. Finally, we will talk about the preventative strategies and vaccination. Unfortunately, there are no preventative public health measures were initiated to reduce the burden of Helicobacter infection or to prevent its spread. Therefore, there is no vaccine available. Lately, much effort has been committed to the development of vaccination. Currently, it has been believed that effective vaccination depends on the induction of humoral and Th2 cell immune response. Thank you so much for your time.